Okay, this mic is on. Yeah.
If you want to walk around, I can get you a lapel one.
Yeah, you want?
Sure. If you've got a lapel mic, that would be great. But we don't have a remote clicker anyway, do we?
Oh, no.
So yeah, it probably doesn't matter then. Yeah. Yeah, I think it'll be fine. Just need it for the recording.
Yeah.
Do you do an introduction?
Yeah. He said, will you take over tomorrow?
I said, you have to, because we can't do anything.
Wow. That's nice.
It's trial by fire. Yeah.
It's great.
Yeah, it's,
Just,
2-3 minutes, or?
yeah, it'll be a few minutes now. I had to force it to shut down.
Oh, you did?
It was still sitting there and spinning. Or had you tried to log into it?
I had, actually, it came back.
Oh, okay. Yeah. I didn't realize. Yeah, just gotta get logged in and start bringing up all the little screens.
It just takes like 10 minutes.
No, this is a fairly new one. I keep it tuned up fairly well.
Okay.
Although it's now dragging. I guess because it was shut down hard. Yeah. Recording's still going?
Yeah. All right, let's go ahead and get started. Welcome back. It's been a month, right? We had one cancellation and then we had 2 weeks off, I think. So welcome back. So before we start, are you guys working on projects, writing up your project? Good. No, that's good. Most of you. I know who's not probably yet. The night before, no. But any questions or issues or problems? Remember, the goal is just to get you to think through a whole project. Don't stress out about it, but we want you to think through a project from top to bottom and kind of go through the motions, because actually putting stuff on paper changes, what your thoughts are, and it should improve the thoughts. Yeah, so email your projects on before the 19th, right? Or the 19th or before.
19th at midnight, 11:59. If it comes in at 12 a.m., I won't accept it.
So email them to Eric or me or whatever. Just, get us a copy. Eric Vidoni, E. Vidoni, Vidoni, or J. Burns 2. So any other questions?
Someone had a question about database access, and I emailed, is that any of you? I emailed you back, I never got.
Yeah,
We can talk about it if you want later.
yeah, it doesn't impact.
Yeah, I mean, some people have a really hard time, so I'm not sure. I have to prove that's. But let's talk about database access and how you would go about first getting the access to the resources that you need and getting help identifying what you want. You want to use something like Heron. There's 100 million, literally 100 million facts in there, and it's not always clear where to find those things. So you can get help from Russ Wake's office. We can talk about doing that. But that shouldn't preclude you from just at least thinking of that through in your project. I'm going to use the Heron database or whatever database.
All right. Anything else? Okay, so tonight we're going to hear from Randy Nudo. Dr. Nudo's the director of the Center on Aging. He's distinguished, has all kinds of accolades, and got a NIH merit grant, right? Which is a really big deal. He's now just recently became the editor for rehab and rehabilitation and neural repair, which is a pretty high-profile journal. So, and he's got an amazing research program. It's very translational. He's a basic scientist doing translational work, and he's going to talk about from bench to bedside and focus on what you're doing or what you've been doing over the years. Correct? All right.
And so I assume most of you are, if you're doing any kind of research, it's human subjects research. There are a few of you that are not. How many, I guess I'll do it the easy way. How many people are bench scientists working with animals? So I knew that there was one in the audience because I know you. But how many of you see yourselves as clinical researchers who may now or one day be doing clinical trials? Okay, and hopefully the rest of you are at least learning about some of the nuances of doing clinical research and the importance of statistics and all of that. What I'm going to do today, I know you've heard a little bit about from other people about translational research and what it is. And I want to give you a perspective from a basic scientist, from the bench level, and how we perceive translational research. Because depending on who you are and what you're doing, whether you're a clinician or a clinical researcher, a clinical trialist, you'll see and define translational research in completely different ways. and your perspective is really colored by that little silo that you work in. So I want to give you a little bit of perspective on this and why we think it's important even at the bench level. So I'll give you some definitions of translational research, and frankly, when I start talking about T1 and T2 and T3 research, if you get confused, how many people have heard of T1, T2, T3? Have you heard that terminology? How many of you are confused by what it is? Well, I am. And every time I read another paper, I thought I knew what it meant, and then I'll read somebody else's paper and they'll interpret it a little bit differently. So there are a lot of nuances. You can go to the NIH website, and Zerhouni, when he put out the NIH roadmap, defined it. The Institute of Medicine has defined it. And so there are all these definitions, and people split it down into finer and finer components. So don't take this as if there is one objective definition of T1, T2, T3, but I want to give you the kind of the continuum of the research process, how basic scientists think about taking basic information and moving it into something that will be useful in humans, and how we should be thinking about doing things the other way around as well. Now, I use a couple of examples from my own research just to tell you how we think about bridging the gap. So when we talk about bench to bedside research, that part is pretty simple. We're defining a disease pathobiology and trying to explore innovative approaches to treatment and prevention, and that's pretty straightforward. But what is translational depends on whether you're a clinical human subjects researcher, an animal researcher, or someone that actually works not even at the whole organism level, but works in cell culture or tissue culture. You ask somebody that works in cell culture, what's translation? I say, well, I want to move my in vitro prep to an in vivo prep. And so it's very far removed from thinking about translating a clinical trial to expanding that to changing clinical practice in general. So there are all sorts of ways you can think about translation. And these are the kinds of graphs that you see traditionally, and I think I think the mouse will work up here. So there are really these three stages, and whatever numbers you assign to them, translating to humans, and actually that's beginning with first in human studies. We're working on a electronic device to repair pathways in injured brains, and it'll be 5 to 10 years before we see the first in human study, and that will probably be a single individual based on some of the FDA regulations. And then it proceeds into phase 1, phase 2 clinical trials, and eventually phase 3 to determine efficacy. And so anything within that spectrum, most people would consider this T1 translation. Now, actually you'll see occasionally when people talk about animal research and maybe trying to find some physiological or anatomical mechanism in an animal model that might translate to humans, trying to find some new target. They'll call that T0. The T1 is really when you're trying to directly apply it to humans for the first time. But then beyond that, the translation, and so for the basic scientists, we rarely even think beyond this phase of actually development of a drug, for example, first in human studies, testing it in phase 1, phase 2 trials, handing it off to a clinical trialist to do the efficacy studies. We rarely think beyond that. That's usually, we consider that the end of the game. But there's a lot that goes on beyond that, and that we'll go into a little bit in this talk. You have a phase 3 trial, and you show that a drug has clinical efficacy, for example, in reducing tumor sizes, well, then that doesn't necessarily mean that that drug is going to be dispensed to everybody that has that particular type of tumor. Does anybody have any? I'd like to hear from you why you think that might not be the case. So, you know, you've discovered some miracle drug that reduces tumor size in breast cancer very rapidly, or halts its progress, and you've gone through a large phase 3 clinical trial that shows efficacy that is better than anything that's ever been out there. So now you're expecting, well, we have we'll spin it off to a company. The company will start producing it, and every doctor, every oncologist in the world will start prescribing it. Why might that not happen?
Side effect profile of the drug?
I'm sorry?
Side effect profile of the drug?
Well, you would hope you would see some of that in the phase 1 study, which is a safety study. So you should know something about the side effects of the drug and its toxicity. So that we don't know. It could still be a problem, but why wouldn't the clinical trials tell you that? I'm sorry?
Because the number of the sample itself.
Yeah, well, yeah, and that's really where it starts. And of course, we can't sample the entire population. We have a representative sample, but are they really representative in a clinical trial? How representative is a sample in an efficacy study in a clinical trial of the entire population? What kinds of things, if you were going to design a drug trial for a cancer study, what kinds of exclusion criteria might be reasonable? The stage of cancer, okay. So let's say, for example, you're going to look at early stage cancer, and you have some operational definition of that, or using standard staging definitions. And so you run your clinical trial on one particular stage. Well, of course, this is usually a continuum, and there's a whole population of people that are along that continuum. And your drug may be totally ineffective in later stages. So that's a good one. So in the clinical trial, you've limited it to a particular subpopulation that may not generalize. Anything else that might be different about the way that you've chosen your clinical trial population? Age? Yeah, sure. That's a great one. In fact, some of the examples that I'll talk about, that's in my field of stroke. That's a very important one, and almost every a trial that's been done in stroke excludes the oldest of the old. And in fact, it's the elderly. Well, actually, today we're actually seeing that the average age of strokes is decreasing because of our obesity and metabolic disorders problems in the younger generation. But age is certainly a significant factor. And it's not that those clinical trials are done wrong in any way, but they are very constrained. And you try to control as many factors as you can in a clinical trial. And not that a drug company would bias things, but they want to see that the drug has efficacy without all of these other extraneous factors. And so a clinical trial, while it can be large numbers, it also can be limited in its applicability to broader populations. So this T2 translation is very important in the further development. Once you have the clinical trial data, that's not the end of the road. Meta-analyses, systematic reviews are needed. Studies need to be replicated in other populations. So this translation to humans is one thing, but then translation to patients, a patient population, is a much broader area. And then even when you do that, there are a number of meta-analyses have been done. The clinical trials, phase 3 trials have been done in a number of different settings with larger populations. Then you come to this problem of translation to practice. Now, even if you find out that this drug that you have will be effective in a broad range of the population with that particular pathology, what kind of barriers would there be to actually seeing that implemented in practice? Why would an oncologist not prescribe a drug that's gone through all of those phases of testing and meta-analyses, a number of clinical trials? Why would they not use it? Well, I'm hoping, think about that one. I'm going to hold that one because there'll be some things that will come up here later that you might think of that that you might consider why something that appears to be effective it actually is not applied in practice. Okay, so these are the Institute of Medicine has defined T1 as transfer of disease mechanisms in the laboratory to development of new methods, more or less what we stated, and T2 from clinical studies into everyday clinical practice and health decision making. From what I talked about in the last slide, that's sort of T2/T3, and that's where the subtleties of how people define these things don't get hung up on it. And the kinds of things you would do in T1 activities, mechanistic studies in cell lines or animals, I would define that as T1 if I'm actually trying to gain some understanding of a pathway or mechanism, a target for a drug for a specific disease state. I would call that T1, and others would put that in T0, that you're not quite testing it in humans yet. So again, those are subtleties. T2, translation of proof of efficacy into proof of effectiveness. And we're going to talk about that difference, efficacy and effectiveness, because they're very, very different things. And adoption in the community. So efficacy is very explanatory, and this is the result of the clinical trial. The phase 3 clinical trials are so-called efficacy trials because they're testing whether the intervention works in people who receive it. And it's very controlled because of our exclusion criteria, inclusion criteria. These are very well-controlled studies. And as we just discussed, it doesn't always include the entire population. So you might, in those phase 3 studies, try to understand how the intervention works, but it's a very tight sample with a fixed protocol. The schedule of and the dosing is very rigid. Now, that is very different from an effectiveness trial, and what people have come to call these pragmatic trials. And so what would be the outcome if this were implemented in usual clinical practice? So I'll give you an example. Here's one that actually was in the literature where they actually specifically did a pragmatic trial when efficacy had been shown. And this was an asthma medication. And this asthma medication had gone through 3 or 4 fairly large clinical trials, very effective in those clinical trials. But when the pragmatic trial for effectiveness was done, which is very different because now instead of these very well-controlled circumstances, you're actually leaving some of the dosing and the compliance up to the individual or the individual's caregiver. So, so you can imagine what kinds of things would be different in, and why would results be different in a pragmatic trial versus an efficacy trial?
Maybe the efficacy trial, you exclude a lot of comorbidities that might affect the outcome, while in pragmatic trials you allow other comorbidities or other confounding.
Sure, sure. So you might broaden the population to other, more greater severity, other things that might be different. Sure, yeah, and you can give them instructions, but it's not guaranteed that they're following those instructions. An asthma medication, they might forget, they might decide they're not going to use it anymore, that you don't have a compliance, and you have no control over that in a pragmatic trial. And but that's the point of the trial, to find out whether it actually it makes sense to use this in no matter how efficacious a drug is. If it has side effects so that people don't use it, then it's not going to be practical. And all of those things, the effectiveness has to be weighed in as well as the efficacy when guidelines are set up to define whether a drug should be used in a certain condition. So these pragmatic trials are actually becoming more common and at least in a formal way. And the design of pragmatic trials is becoming a science in itself. And they're very important in that translation from the efficacy of human studies into practice. So we usually think about this translational pathway as moving from basic discoveries in a one-way fashion to mechanistic studies, initial human testing, proof of efficacy, proof of effectiveness, and then diffusion to various types of healthcare settings. There's a term that's becoming more common, something called reverse translation. And reverse translation refers to the fact that a lot of information from especially these early human trials, is used to decide how the basic science should be done. And in fact, this is very important, and this is one of the things that I do very frequently, is start with the clinical question, and rather than starting with some esoteric molecular pathway in an animal model and deciding how I might use it to design a drug for particular pathology, actually taking a human condition such as stroke and learning from the clinical trials what questions need to be addressed in the animal studies. We don't do that often enough, and this requires a lot of work to get people to really collaborate on these kinds of studies in a true translational way, because our language is different. We define terms completely differently. So it requires a lot of work and time, but more and more I think we're going to see that these teams, interdisciplinary teams, are going to be most effective. And I think the more that you buy into this notion, the more marketable you will be if you want to do research, either at the bench or at the bedside, if you understand that your role as a translational scientist is really going to be where the help propel the next wave of discoveries in any of these areas. So I want to just talk about this just because I think you can learn a lot by how people define their role in translational research and how they think about it. And there was a survey that was done a few years ago at Johns Hopkins on faculty involvement in translational research and what type of translational research they see themselves as doing. And it turned out they got 1,800 people among the faculty and postdoctoral fellows to participate, which is amazing in itself. 55% of the faculty and 40% of the postdocs actually responded to this. And they did this across schools of medicine, public health, nursing, and engineering. So that's another aspect of how people in different cultures, and these schools truly are different cultures, how they see their role in translational research. And the survey was actually done in 2008. And I'm just going to highlight a few things from this table, which it's easy to gloss over, but just to highlight a few of the points that came out of this. The people that, out of that 1,800 people, 69% said they participated in some way in translational research, which is, I think that's reasonably high. Most of those were consider themselves as doing T1 research. Again, that would be either animal research or moving the findings from animal research into human studies, early trials. Those were all T1. Relatively few in either T2 or this reverse translation. It's actually not too surprising, but there was no significant difference between males and females and whether they're doing translational research, although there was a little bit higher prevalence of T1 research among males, but not a huge difference. Whether you're an MD, PhD, or MD, PhD didn't make a huge difference, although the PhDs there are actually fewer of them that did not consider themselves as doing translational research. I find that a little bit disturbing myself because I think that more basic scientists need to think about their role. These days, grant funding is so hard to get. It's so competitive at NIH to get grants that, and you need to justify the rationale for what you're doing. You can't do some basic esoteric scientific research without a clinical application and describe its significance. That it's actually a little disturbing to me that there aren't more PhD trained scientists thinking about translational research. So, there's actually a lot of thought now to whether among bench scientists that while it's so hard to get funded, maybe I ought to become a clinical researcher. Well, it's actually a pretty steep learning curve if you're trained in bench research to move into clinical research. It can happen, but but I think all of us need to think about collaborating with those individuals. If you're a clinical researcher and you want to do something novel, talk to somebody that's a little bit outside of your field. Talk to a PhD and level scientist about your particular clinical problem, how they think about it. You're going to think they're off the wall because they're thinking of these esoteric questions that have little clinical relevance. as you begin to develop a dialogue, I think inevitably you come up with a new perspective, and both of you will come up with a new perspective. Whether you're at a fellow at a junior faculty level or a senior faculty, did seem to make a difference. The fellows did much less translational research than the faculty. Any ideas? Because there are probably some fellows here. Any ideas why that might be? Pardon? So why would that? Because you're moving on in a year or so. Why would that affect whether you are doing translational research? Yeah. Well, any other opinions on that? I mean, what about the? I mean, if you're a clinical fellow, and I assume that most of your time is involved in clinics, you don't have time for much of any kind of research, and if you do, it's probably going to be directly related to your clinical practice. Is that valid?
Because the research is a process, it's going to take time. So even though there's limited with 1 year, it's still limited with what kind of research you're doing. So if you are a fellow, you're limited with that amount based on the criteria for study that you're doing.
Yeah, okay. Whether you're in the School of Medicine, public health, nursing, or engineering, this actually makes a pretty big difference. You can see that nursing and engineering do very little T1 research, and engineers do almost no T2 research. While the bulk of the School of Medicine is doing T1, and nursing is primarily doing T2. So that's actually kind of interesting because I'm not going to answer this today, but think about engineers and what role they might play in moving the results of basic clinical trials into practice. I mean, you might think that there's no role for an engineer, but the engineering is a huge field, includes bioengineering, and I think that we need to, when we see these gaps, we could probably capitalize on how those different fields could be used.
Did everybody in this, so maybe I missed this, did everybody feel like they knew about translational research? Because I wonder if when you become an assistant professor, you start learning the lingo. I should be saying, I do translational research in my grants, right?
Yeah.
And engineers, maybe that's not the lingo. And did everybody in this survey feel like they knew about all the...
It's not clear when you read the methodology whether that was, how well educated they were about the terminology. And, I think they defined it for them, but how well they grasped that, I'm not sure. And that could very well be part of the issue. And again, I think learning about this development and evolution from bench to bedside research early in your career will be helpful, I think, in moving some of these people along earlier. Teaching people in engineering about translational research. I think when you see a shocking number like 9 people doing T2 research in engineering, it looks like an opportunity to me.
The residents in here, I know your guys' projects are all at least somewhat translational. Maybe you did or didn't identify it as doing translation research, but all your projects are to some extent. And so it surprises me that fellows rate that so lowly,
Yeah.
especially if they're looking at their own clinical practice. That really is.
And some of this is actually, you might expect, people that are basic researchers are primarily doing T1 research. Clinical researchers, actually, it's split almost 50-50 whether they consider this being T1 or T2. Population public research, as you would expect, would be more T2. And engineering, while there aren't that many doing translational research at all, most of that is in T1. So it's interesting to think about how these different fields, and depending on where your degree, your rank, what school you happen to be associated with, really does affect where the opportunities are to do translational research. And so when you looked at some of the reasons why people did not do translational research, the biggest one was it's not part of my research agenda, not central to the goals of my research program. Well, you know, it's at some level, many of us feel that it really is part of any medical research program of any kind, is helping to translate that information. Even if you're doing clinical trials, if you're doing a clinical trial on something that is never going to see the light of day in clinical practice, maybe because it's too expensive. I mean, Dr. Benoni could probably tell you about some of the rehabilitation trials have been done and how many hours a day have to be spent in therapy, and they show efficacy, and yet, you know, you calculate those hours, and, you know, if you have to spend 5 hours a day, 5 days a week with a therapist for 6 weeks, nobody's ever going to do that in practice. So everybody needs to think about the long-range prospects for an intervention to actually be used in practice. So some people are not sure what the next translational step would be, and that's just, you know, because we don't know about the process. Haven't identified an appropriate translational research question, and don't have the training or expertise to make the work more translational. So most of these, I think, have to do with, or haven't identified or secured colleagues or collaborators to make the work translational. And again, I think we just need to develop, and I think it's happening more and more, our CTSA is geared towards that, to create an environment where translation is a focus. I think most of the people in this study agreed that translational research is necessary to improve health outcomes. It's professionally fulfilling. The scientists who can cross the lab-clinic divide are in demand. There was a large feeling that that's the case, and I think in the funding climate the way it is, that's going to be true more and more so. Some people are a little bit afraid of the regulatory requirements in translational research, but many see that it's under-recognized. And I think my eyes are starting to glaze over by all the graphs, so I'm going to, yours probably are too, so I'm going to skip that one. So just to summarize this, we really do have this gap in translation. We have basic scientists working on their own and trying to identify new targets and understand the etiology of a particular disease, and but don't understand the clinical environment and the environment of clinical practice. So these are things that we really need to develop and educate each other about to really be more efficient in this whole process. So I want to just talk about a couple of examples of how these things work or don't work, as the case may be, and where things go wrong when you try to take information from basic science, develop a new intervention, and tested in a clinical trial. And these are just a couple of examples in my field of stroke. And, you know, these are just the demographics. We have about 800,000 new strokes each year. And with an aging population, stroke prevalence doubles by decade over the age of 50. And kind of interestingly, in the younger population, there's a greater prevalence in women. Men catch up later in life, so it turns out, it comes out in the wash, but it clearly age is a significant factor. And with the growth in the population, we expect this number to rise. It may have leveled off over the past few years, but we still have a lot of aging baby boomers. We have a lot of metabolic disorders in certain parts of the country. It's terrible. I was just at University of in South Carolina and in Charleston, and they were talking about the young population of stroke survivors that they have, and that the figures they have on obesity among the young are something like 20 to 30%, depending on the survey, of young people around the age of 20 being obese. I mean, it's really a bit frightening, and so these are going to be associated with additional strokes, and strokes in younger populations that we don't completely understand because we haven't studied as many of them. And so someone that's just coming into this field and thinking about what they might study, they could be studying prevention, awareness, treatment, and certainly prevention and treatment are important. The American Heart Association thought that the way to prevent strokes is to make people aware of risk factors, and so we know what they are. I mean, this is pretty straightforward, very solid evidence on risk factors: hypertension, smoking, physical inactivity, diabetes, alcohol intake. All of these are clearly associated with stroke, and collectively, all of those risk factors account for nearly 90% of stroke. So this is something we can really do something about. The American Heart Association was really intent, and they still are. About a few years ago, they had a big push to reduce stroke deaths with greater public awareness. And this is sort of a different type of research. You could, how much effect can you have on a population simply by making them aware of risk factors and signs of stroke. And so back around 1996, they had a big push, and this was the map of stroke deaths across the U.S. And there's this big pocket in the southeast, the stroke belt. There's actually a stroke buckle in that South Carolina area that I told you about, where rates are double the national rate. And if you look at the numbers, the age-adjusted rate in the red was 30 and over. And that was 1996. And so they had this big push, teach people that that strokes are an emergency, that they started calling them brain attacks. You have a limited time, get to the hospital quickly. And they had this acronym FAST for face, arm, speech, and time to learn the signs of stroke. And then about 10 years later, they looked at this map. Map looks about the same. There's a few changes, but now if you look at the numbers, I don't think you can read them. Instead of 30 and over being the red states, it's 50 to 60. So the death rates per 100,000 population almost doubled in that 10-year time frame. So despite their program of awareness, it didn't really have much of an impact on death rates because of other factors like age overshadowing this effect. So we know quite a bit about outcomes after stroke and people with either minor, moderate, or severe strokes. If you look at this is just as one of the many measures of motor function. People recover spontaneously, and there is some leveling off of recovery after 3 months or so. But the most important thing is that is time, and if you intervene early, either with a clot buster like a thrombolytic agent like tissue plasminogen activator, or there's some mechanical devices that can remove clots, you actually can have a significant impact on how well people recover and whether they survive or not. The problem is that thrombolytics are only given to about 4 to 5% of stroke patients. I don't know how many of you have experience with acute stroke, but why such a small segment of the population when we know how effective TPA is?
There are a few things. The time, I guess, when the patient arrives, the patient cannot get therapy any time, and even now, each person is different than the other. Each patient, so you have to have good CT scan in order. Like, I mean, if you can get it even within half an hour, but the CT scan is bad, nobody will give that's one thing. The other thing, I guess, that's include.
Doctor level physicians, like neurologists are not everywhere. There's so many people out there who do not get to see a neurologist who have a stroke because they are physicians are not comfortable.
And I think that's include major stroke. I think that this number include minor stroke, and minor stroke they don't get TBA anyway.
Yeah, that's actually a good point. Yeah, all those are good points, and really encompass the reality that the time between the event and entering the hospital, and then the so-called door to needle time is that could be quite long. People often aren't aware that they've had a stroke until long after the fact. They may have it in the middle of the night. This happened to actually, and one of the things about not having a neurologist, people like my mother who are. Yeah, it's becoming better. I think if you live in an urban setting, you're lucky because you have access to major healthcare systems. But if you live in a small community or a rural community, you're out of luck with stroke. And my mother was a good example. She went to a small community hospital that actually was the next morning because someone convinced her that her speech was slurred. And it was the weekend, and they had no neurologist on staff. They had no radiologist on staff. And they said it'll be Monday before we can have this read. And this is a small community hospital. It wasn't terribly unusual. And so fortunately for her, she had a TIA, and it was she recovered just fine. But you have to, the drug is not effective. You have to do the CT scan. It's not effective beyond 4 to 5 hours, and that number is pushed a little bit depending on the clinical study that's been done. CT scan will rule out those with hemorrhagic stroke. Those are only about 15% of the strokes, though. So you have 85% of the people potentially could be eligible for TPA. If they get there early enough. But you have to know when the event occurred, and it has to be within that 4-hour window. So 4.5 hours was actually from a European trial where they actually had a slightly different criteria for entry, but it certainly it won't be beyond that. After a certain period of time, beyond 4.5 hours, you actually can cause hemorrhagic transformation with giving TPA. So it has to be confirmed to be ischemic and not hemorrhagic, and hemorrhagic transformation can occur. So from the basic scientist perspective, we are all trying to develop a better drug to protect the brain after stroke. And so there are all these, we know, all this is just a summary. We know everything there is to know about the pathways that lead to brain infarction, excitotoxicity, inflammation. So glutamate, NMDA receptors, calcium influx, free radicals. So any of these could be potential targets. Develop an antagonist to NMDA, an enzyme inhibitor to nitric oxide, and so on and so on. And so neuroprotective agents were developed by the pharmaceutical industry in phase 3 trials. There are a large number of them. There have been none of them that have been shown improvement in clinical outcome with neuroprotective agents. And and this was actually from a few years ago, over 115 phase 3 clinical trials, over 1,000 trials at other phases that failed. So just developing a drug that affects a and seems to work in a rodent model of stroke, the pharmaceutical industry spent billions of dollars, literally, in more than a decade trying to find something, and we still haven't found anything that's that has efficacy in reducing infarct size or improving outcome. So what the community did was get together and had a big workshop. I was in the first of these workshops to decide how to approach this problem. The Stroke Therapy Academic Industry Roundtable, people from NIH, people from academia, pharmaceutical industry. And so we developed a set of criteria and said, okay, in order for something to go into clinical trial, before we do, the preclinical studies have to meet a set of criteria that will make it more likely for these drugs to be effective, and before they will enter human trials. And I'm not going to go through all of these. There's a host of them, but some of the ones that actually are important is the time window. Time and duration has to be appropriate to the clinical protocol. The larger species need to be used, primate species especially. And so there were some drugs that were developed according to these criteria, and AstraZeneca developed this drug that was a nitrone derived compound that had free radical trapping properties. And we thought, okay, we finally have a drug. It worked in rodents, it worked in primates, and then they actually worked, it had rabbit models, improved recovery in primates when given 4 hours after permanent focal ischemia. And I won't go through the data, but the clinical trials, they had first phase 3 study, and it showed reduced disability on a common outcome scale when given within 6 hours after stroke. NIH stroke scale, which is a little less sensitive, didn't show any change, but people were pretty excited. We're on the right track here. So there was another phase 3 study, almost twice as many patients, and it failed to confirm efficacy. And so that was problematic. So now everybody starts arguing in the field, and that some said there were possible impurities in the first clinical trial, and the impurities actually caused the benefit. I mean, that was probably, that sounded a little crazy. That the STAIR criteria were not really met in the primate study, or that the criteria still aren't adequate. And so, there were people back and forth arguing with each other, pointing fingers, the clinical scientists pointing to the basic scientists, and vice versa. So I won't go through all the problems, but someone brought up earlier age, and nearly all animal studies in stroke utilize young, healthy animals. And in contrast, humans with stroke are usually elderly. They have other comorbid conditions like hypertension, diabetes, myocardial infarction. There are a lot of people in the scientific community that feel that our animal models simply aren't adequate, and we need to have animal models with these other comorbid conditions. We'll never create the perfect animal model because most laboratory animals don't have spontaneous strokes. I've been working with monkeys for about 30 years. I've seen one out of probably 2,000 that have had a confirmed spontaneous stroke. And so you're going to wait a long time before you actually have that happen. So we have to create experimental models. They're not perfect. There was another study, another approach with electrical stimulation, epidural electrical stimulation in stroke that worked very well in animal models. And I actually was part of a large multi-site preclinical study that NIH funded to test this. And it worked very well in animals. There was a company that was created that produced an epidural stimulator that was used in conjunction with rehabilitation. And they had a series of trials. These are pretty small because when you're implanting devices neurosurgically, you usually can't use the numbers you would in a drug trial. So they're necessarily small, but the early studies actually showed some pretty promising effects with the investigational arm and the control arm. And then they did their phase 3 study, and almost about 31% receiving the cortical stimulation and the rehab therapy, they reached their defined threshold for clinically meaningful improvement. You look at the control arm, though, and 29% receiving just rehab alone reached threshold. And now, this was a publicly traded stock at the time. I didn't have any stock in it, fortunately. I was an advisor for them, but I didn't have any stock. They didn't take my advice either. So their primary clinical endpoint required a 20% difference. They were way off. And this is what happened to their stock the day they announced that those trials at the American Heart Association International Stroke Conference. Their stock went from about $12 a share down to $2 a share, and they went out of business about a year later. Now, that's one trial, but those of us who know why it went wrong, and it probably should have worked had they done the trial correctly. But it just points out that there needs to be a lot of communication between the basic scientists and the clinical trialists. And when industry gets involved, there are certainly other issues that are driving how those clinical trials are being run. Not that they're necessarily right or wrong. We're involved in this right now, trying to decide how to get a surgically implanted device developed out to market and into human patients. We can't do it. I mean, when you look at the cost that would be involved in developing that product through a first phase 3 clinical trial, it's about $70 to $80 million. So we can't go out and raise that from NIH. You've got to form a business. Only business is going to have the capital to drive those kinds of trials, and they have an agenda. I mean, if they're a business, they have a responsibility to their shareholders in addition to the patients they're trying to serve. So this really is an issue in our current climate. That no matter how good your intentions are in developing a translational research team, at some point, it's got to, this is part of the practicality. Getting it out into the marketplace is more than just doing that first phase 3 clinical trial. It sometimes involves how the business community reacts to it. So just and I'm ending now. We can all go out into the cold weather, so you might be happy to stay here for a few more minutes. The prevention, awareness, acute treatment, chronic treatment are different areas that one could be examining in this field of stroke, but certainly we need more interaction if we're ever going to make inroads in some of these very difficult disorders. We really need to have teams working together towards really common questions. So I'll end there, and if you have any questions, comments, I'd be happy to discuss with you. So now that you know what translational research is, how many people, if they were to be doing research right now and you have your first NIH grant to do it, would you be doing, how many would be doing T1 research? T2? T3? None of the above. Maybe I didn't mention T0 or T4. Those are some new ones you'll hear about too. We use all kinds of numbers. All right. Well, thanks. I hope I gave you a little bit of perspective on from the scientist's point of view. Thank you very much.
Class next week. I think we have one more class. Hey, I got a question for you.